So before we start the talk, we request our head of the division, Dr. M.S. Sekhar, to say a few words.
So, very good morning, madam.
Good morning. Good morning.
On behalf of SIBA, Director SIBA, and Dr. Rota and other participants and head of the departments and scientists from SIBA, we welcome you, madam, for this Antimicrobial Awareness Week. We are blessed that, and we are also fortunate to have you here because the expertise which you have, I think you are the best amongst the aquaculture scientists to give the awareness, especially for the younger generation. And we know that in Awareness Week, madam, the theme is preventing antimicrobial resistance together. So the together is the word which I feel together we can solve this problem. So guidance from your side is always there, but how to implement this use of antibiotics, especially in field of aquaculture, little strictness is required. And we know the policies and the guidance and other directions exist, but it is slightly difficult at the field level. And probably we have to we have to now generate more awareness, the restricted use of antibiotics, especially in aquaculture. So with this background, madam, I once again welcome you. And madam, as you all know, she is a renowned world aquaculture scientist, having expertise in disease diagnosis, disease management, and other aquaculture practices. And antibiotic resistance, definitely, madam is also is a renowned figure in that thematic area. So with this few words, madam, I now the forum is open, and kindly I request you to kindly give the presentation. Thank you.
Hello, is my screen visible?
Yes, madam.
Okay. So, first of all, I would like to thank the organizers for this opportunity to speak on Antibiotic Awareness Week. Now, during Antibiotic Awareness Week, as you would see, the importance is realized from the fact that it lasts over a week when we are trying to address issues related to antimicrobial resistance and its awareness. Now, if we look at other important days that are celebrated, it is usually, say, Father's Day, Mother's Day, Environment Day, and so on. But in the case of antibiotics, it is called as a week. But like all days, all the year round, we need to be aware. But the fact that this extends over a week implies that it is more important than all of the other celebrations during the entire year. Having said that, I would like to now thank Dr. Shekhar for his kind invitation, and I endorse his views that education is the key, because no problem can be solved without recognizing it, and also without making everyone aware of it. So when it is a problem that has to be addressed by every individual on the planet, it really means that solving is not going to be that easy unless there is a concerted effort on everyone's part. Now, when I was asked by Dr. Rota to speak on okay, antimicrobial resistance during this week and to choose a topic, I knew AMR and One Health, alternatives to antibiotics, and all. I've been speaking about this time and again, and your own institute during the last year's celebrations, I had spoken again. So I had to make sure that I am not just giving a talk that is a repetition of whatever I've spoken. So each time, it's like having an old wine in new bottle and presenting. So I thought, why not we talk about what is happening in the environment, the dissemination of the problem. So we know that One Health, the word One Health encompasses human health, animal health, plant health, and of course, environment health. So organizations that have been involved with, say, human health, like the World Health Organization, the Food and Agriculture Organization, then you have the World Organization for Animal Health, who are formerly known as OIE, all are jointly working towards the same goal of One Health, but now it has become a quadripartite agreement with the UNEP becoming also a part of it. Now, when we scan through the newspapers, we invariably see some news or the other that attracts a lot of attention. This appeared yesterday, where it said 70% of the antibiotic formulations in the market in 2020 are unapproved. Now, when we look at this kind of information, we know that we have a lot of regulatory bodies and approvals are necessary, but these kind of spurious drugs, they actually come in the way of bringing about more resistance and making infections very hard to be treated. And therefore, whatever rules can be made, whatever regulatory approvals are got, it is very important that we have education, and when everyone participates in it, as we saw during the COVID pandemic, where every individual, I think, became very PCR educated and knew the word RT-PCR when scientists thought it was such a very glorious word only to be used by the scientific community. So we will be able to tackle problems only when we look at them. Now, antimicrobial, when we say yes, it has become almost synonymous with antibiotics, which we will be talking about. But then we know these problems get, I should say, more serious when you find them in association with other pollutants like pesticides. We have heavy metals and many other kind of molecules that find their way. You may be using, especially in the context of aquaculture, we know very well, but it's used in all sectors. So if we have a leather industry and we have chromium coming out of it into the water body, how does it make an organism even more resistant? So we need to study that as well, and we cannot overlook all the other pollution that comes, and treatment of wastewater would therefore have to be addressed in a holistic manner. So this again yesterday appeared in the Sunday's newspaper, where there was an article on pesticides reigning supreme in Indian farms. Now, why am I talking about agriculture or about, as we move on, about poultry? We will talk about livestock and also, of course, about industries that are polluting. All of this are going to contribute to the water and making it wastewater. And when aquaculture is performed in those environments, we know the problem gets many-fold. And therefore, we need to understand that wastewater treatment or looking at aquaculture in particular waters would also be contributed to by, I should say, biomagnification of a large number of very serious agents in food, in fish, and during the culture process. Now, WHO has released list of world's most dangerous superbugs, so medical is no exception. We need to know all this because is aquaculture always only a culprit as is projected, or is it a victim also? Because water body is something that is common to almost all the industries that have to let out their effluent, and one cannot have borders where the water goes and reaches. So there are, of course, critical antibiotics and priority 2, which are highly important, and all of us are also familiar with the escape group of organisms which the WHO has enlisted. Now, when it finds its way to the water through whatever sources, the hospital effluents, then or from the pharmaceutical effluents, they all contribute to the problems that we face in aquaculture as well. Now, some publications of the past had raised serious concern, and things are getting manageable, but not yet as much as it should be. So heavy use of prophylactic antibiotics in aquaculture was projected in a very big way, but antibiotics for prophylaxis is no longer being used, and this problem has been managed, but it is there in other sectors, in the livestock sector, in the poultry sector. Yes, it's used. So aquaculture really becomes another environmental gateway to the development and globalization. But I, for one, would not like to think that aquaculture is all the time a culprit in the antibiotics that are being used, but more often it is a victim of all the other sectors where water becomes the source for being used in aquaculture. Now, a global antimicrobial consumption in all of these various sectors clearly shows, we have seen, all of us have seen these slides again and again, and we see in fish it is minuscule, but look at the volume used in piggery, in poultry, in the human health, or in livestock. So this is why I'm convinced that aquaculture can be helped, but all the other sectors have to help us to help aquaculture. So between animal and human medicine, there's really no dividing line, and it was, the word One Health really was very, very long ago pronounced, and I also find myself someone who has been working in the area of One Health very much, because I started off as a medical microbiologist, then was working in a college of fisheries where it was, a veterinary university associated work also with other samples like livestock, poultry, and then, of course, now back to an institute, a health university where environmental problems are ruling the roost. So Dr. Rudolf Virchow was a physician, anthropologist, pathologist, prehistorian, biologist, writer, editor, and politician. So all in one, who showed what One Health means, and who also helped us see that you cannot have walls that divide between the different subjects, and you can see the importance of whatever you are doing in relation to every other sector as well. One Health is important because, of course, this is a CDC slide. See, the whole world is undergoing, I should say, cardinal changes. Population has increased, and therefore the number of people per square meter will naturally go up. We know about fish, we talk about it all the time, but this happens even with human lives. Climate change has been very important for some of the habitats changing and the populations moving from one place to another. A lot of travel and trade and the transportation of live animals, all that has come in the way, and the use of animals more than just for food, pets and the hobbies that people have all been responsible for the spread of diseases between animals and people. So the One Health actually takes into consideration human, animal, plant, and environment. Now, One Health issues include, of course, emerging, re-emerging, and endemic zoonotic diseases, the neglected tropical diseases, vector-borne diseases, antimicrobial resistance, food safety and food security, environmental contamination, climate change, and all the other health threats that are shared by all the people in the sectors. Now, so as Alexander Fleming, when he discovered penicillin, of course, it was a very, very wonderful discovery where he said never to neglect an extraordinary happening, but his foresightedness and his predictive ability made him see that an evil was waiting to be actually leading to resistance. And he calls the thoughtless person playing with penicillin treatment being morally responsible for death of person who succumbs to the infection. Now, this is what is happening in today's life. Now, this book beautifully, of course, shows the rise and fall of antibiotics with the impact. It's a predictive one. So by 2030, by 2050, and by 2050, how much is the economic loss, how many deaths per year would happen, and what is the volume of budget that would be spent on actually daily dosages of antibiotics, that would be decided, So now the problem with AMR runs deep in every country. It's not only India. That is, so higher the income or higher, or when I say higher the income, advanced countries. So antimicrobial resistant genes, ARGs as we call them, the bacteria that carry resistance, the wastewater treatment plants or the urban water system, all of them have to be addressed in every country. Some are more advanced than others, but lessons to be taken from one country and how they operate is very important for those who are still at the stage of deciding how to go about. Now, a cursory glance at the use of antibiotics and the kinds of organisms that are encountered in all these countries is just presented. Now, in India, yes, you can see it looms large, the problem, because the number of bacteria, the kinds of bacteria and the antibiotics both are presented in the graph. But it is not very surprising considering we are a very populous country, okay? So I don't think we are overdoing anything, but at the same time, we need to do better. We need to understand better the problems. So like we talk about other health conditions where the do's and don'ts are always advised, I think aquaculture is no exception, or for that matter, all the other food producing sectors, food security, nutritional security sectors, the pollution management sectors, all of them are very important. So there are many ways in which, of course, we have recently come out of a pandemic. We know there are many infectious diseases that exist, but the communicable diseases such as diabetes and cancers also rule the roost. But somewhere there you can see antimicrobial resistance is predicted as the next pandemic. And if we look at this timeline, you would see the antibiotics that have been introduced and the antibiotic resistance that have been discovered go hand in hand. Now, the golden era was from about 1942 to 1960 with the discoveries of many antibiotics. But we have come to a point where there is a discovery void. And now in the last about 2.5 to 3 decades, there hasn't been any new antibiotic discovered, which is a cause of concern. So historical timeline of aspects of antimicrobial resistance at the global level has made the recognition of the problem global. So while some of the intergovernmental organizations got together by about 2010 and founded the tripartite leadership roles for AMR governance, India didn't lag behind. And the National Action Plan in India was also prepared in 2015. And now if you look at 2022, it has become a quadripartite program. But, there are, of course, a lot of funds being generated, because if we talk about identifying AMR, a lot of funds have to go into also methodologies. A lot of funds have to go into developing rapid diagnostics. Now, if we look at antibiotic usage by any clinician or by any aquaculture farmer or in any other sector, one is not patient enough to wait for 48 to 72 hours to have the results in order to decide what would be the best that could be given. Because empirical treatment is the order of the day. If I'm sick, I would rather have someone treat me today based on the clinical science rather than wait to see after 72 hours the best treatment. This is because many signs are common to many disease conditions. So if I have cold and cough, it could be viral, it could be bacterial, it could be allergic. Just like how if we see hemorrhagic lesions in fish, it could be due to a virus, it could be due to bacteria, it could be due to a protozoan having caused a problem, or it could be merely a traumatic experience that has happened in the fish. But one doesn't wait and dumps a lot of antibiotics, and that's where the problem really begins. And therefore, in order to address antimicrobial resistance, diagnosing them very early is very important, and in all kinds of samples. And more than diagnosing after a problem has occurred, can we have prediction, preventative measures that can happen? Okay. So, in short, if you see, the environment is really a repertoire of the waste that comes from every sector. And the interrelationship of all the sectors is something we need to remember. We know when water is the environment where all the pollutants find their way. We know that anthropogenic waste from homes, domestic use, everything that finds its way, it may have detergents that carry toxic chemicals, pharmaceuticals will release a lot of antibiotics, and the germs that come from bacteria, particularly that come from all the other sectors, would also be related to each other. For instance, see, the poultry gets fed by a lot of fish meal also that is used, and likewise, we are aware that waste from one sector is sometimes a food for the other sector. So where do we start looking at control measures? That can happen when we have understanding and treatment that is done to any sample before it becomes available for use in another sector. So, of course, some of these figures are all from our own papers. So hospitals, we can see how they select from the patients, say, vomiters, or the sewage material that comes from the hospital, or the waste blood, the waste drugs that are thrown away. All of them find their way to the wastewater treatment plant. Then agriculture, I already mentioned in my first thing when I showed you about pesticides being used and antibiotics being used. They also find the water that is used for irrigation finds its way to the groundwater, or also to the river systems if they are on the banks of rivers. And in countries like Israel, we know they're all, there's a lot of only saline water agriculture that goes on, but they manage very well, which we will just see how different countries manage. So transfer from livestock, farm to fork perspective needs to be kept in mind. Chicken, which is a hot favorite of many of the non-vegetarians, also becomes the source of so many antibiotics that are used, and consequently the resistance to the bacteria that gets transferred, not just to other pathogenic bacteria, but to the innocuous bacterium. It could just be the microbiome of the environment. It could be the microbiome in the human body, and community-associated selection of this resistance takes place because this transfer within the human gut happens through horizontal mechanisms. So what really happens is all the antibiotics generally kill commensal bacteria, while the resistant ones tend to survive. And when they multiply, they also spread between animals, humans, and the environment. Well, we did some work about 4 decades ago. Even Dr. Otta was involved, and there were many young scientists who are now in many important industries. Dr. Srinivas Rao, who is presently with Novartis in a very senior position. Then we have Dr. Dhirendra. And all of these people worked on adding the different chemicals that were used in aquaculture, innocuous chemicals, or just to clean up the environment. It may be BKC, it may be TCK. And whenever they used other antimicrobials, like even sanitizers in the water, they found that all of the organisms that was participating in the biogeochemical cycles were destroyed, while other bacteria like, the autochthonous bacteria like Aeromonas or Vibrios survived very well. And this result was even more conspicuous when sediments were used in this study, because sediment really becomes a very protective environment where the organisms formed biofilm survive. And a temporary, I should say, quiescence when you do a testing by conventional methods, which makes you feel euphoric that you have controlled the pathogen, it's really very short-lived, because within 48 hours, the numbers of these organisms rise very rapidly once these chemicals are withdrawn. So this is, of course, a pathway map of the antimicrobial agents as well as the resistance. Now, so what happens when we use antibiotics so much? Novel strains emerge, they limit the efficiency of therapies. Then traditionally, attention has been singularly targeted towards clinical, veterinary, and food-producing animal settings. But environment today has been increasingly recognized as having the most significant role in the development and spread of AMR. Horizontal and vertical transfer mechanisms between all of these sectors that were discussed before keeps happening, and many mobile genetics are involved in transferring the drug resistance. So what is the purpose of monitoring, say, antimicrobial resistance in the environment? Now, the goal of sewage surveillance, because that is one thing that is predicted. We always wonder, how can we do it? Predictive models are being prepared because it's an everyday affair and it is happening from all sectors, and whether it's urban or rural, there is, of course, a lot of sewage material finding its way to the environment. So the goal is not directly related to the risks that are associated with AMR, but the purpose is to use sewage as a bulk sample of the human population in a confined area. And you will realize that the, I should say, the mineralization or the lack of AMR being present in the rural communities is much less. I mean, the AMR is less as compared to urban areas because of some of the mechanisms that we will discuss. So considering monitoring as a tool, we need to assess risks to human and animal health with environmental development and transmission of resistance. So it is fundamentally important to take into account the types and roles the environment plays regarding AMR. So the questions that we all, I think, that comes to our mind is, what is the purpose of monitoring? What would be the choice of methods? What are the targeted environments that we should take? Who are the intended users? Integration potential of all the sectors. Now, we do hear how in the UK, even sewage, the water is processed 7 times before it is completely potable. And 7 times that water is used before it is discharged. Now, the very thought that we are actually taking very high quality potable water from the taps in the UK is sometimes amazing. And how they do, what they do, these are lessons that we need to learn, because the amount of water that we use in our country is truly much, much more than what they use. Because water is the elixir of life world over, but for India, it's even more with our weather and with our lifestyles, where instead of a shower, which would use, I think, 1/10 the water that we actually take in a bucket and bathe, the quantum that would be there as waste would be much higher. So monitoring in the environment, when we have to do, where to watch. Now this encompasses, okay, urban, rural, different processing sectors, hospitals, and this lifestyles. Open defecation is something that is there in the villages, but fortunately, because of the vast areas available and the less populous areas in the rural sector, it doesn't become as much a threat as in the populous areas in urban settings, where processing that sewage or other anthropogenic waste, as I said, it could be the detergents used in the house, all of them find their way. Now the processing plant, because as a country's, I should say, status improves, more and more of processed materials becomes available. So you get processed fish, processed chicken, processed beef, processed mutton. And in all of these processing sectors, what happens is, with the waste from the processing plants finding their way to the water, how is that pollutant going to be taken care of? About industries, less said the better. We are not even at this moment talking about too much of the air pollution, because even the air pollution from the industries comes back in the form of acid rain to the water environment, which is why we see sometimes aquaculture, being affected because of places where there is acid rain or where there is acid sulfate soil. So the sectors are so many, and problems therefore are multidimensional, affecting the economies, the environment, the social, human, and animal health. And AMR is prevalent in all of these associated places, the water, soil, people, animals, food. So water becomes the important link everywhere. Having said that, in the wastewater treatment plant, the resistance mirrors the pattern of antibiotic resistance prevalence in any environment, whether it is in clinical or processing areas or what is used in food preservation. So these wastewater treatment plants, they contain antibiotic residues, the bacteria, they contain a lot of nutrients and other selectors. And I mentioned a little bit before about the selectors, such as a lot of heavy metals or other antimicrobial agents that get to be used, disinfectants that may facilitate the interactions leading to genetic transmission among bacteria. So AMR in wastewater includes many classes. So you have antibiotics like penicillin, tetracycline, quinolones, macrolides, aminoglycosides that have been detected. And the sewage contains, of course, the bacteria, the genes, and common pathogens, which are major drivers of AMR. And this includes the escape group of organisms, which is a worry because it comes back to the humans, apart from all the pathogens that may be present, bringing about problems in the different sectors. We will be, of course, briefly looking at how the wastewater treatment happens. We are familiar with it, the primary, secondary, and the tertiary treatments. But look at the final treatments, which, if we undertake, would render it free of a lot of antimicrobial bacteria, yes, but also the genes, the nanofiltration, ultrafiltration, microfiltration, the reverse osmosis, and of course the chemical processes such as photocatalysis, phenton-based, ozone-based, and the electrochemical oxidation. But we will see the disadvantages of some of them as we go on. Due to paucity of time, I will just rush through some of them. So the question comes, now I have added one box to all of what is done. How do you monitor? Now we are aware that many times the bacteria may be absent, but the genes will be present, and therefore qPCR becomes the order of the day. And when we use qPCR, if we have to use either the culture technique or the metagenomic approach, we would get an idea of the quantum of resistance that is going to be present. Now, if we look at, this is taken from Yes Encyclopedia, but if you see the primary treatments and the secondary treatments, if very effectively done, do come to a point where your secondary effluent, when treated with disinfectant, will allow this, at least the generation of a quality of water that can be discharged into the environment to be used, maybe for agriculture purpose or for other purposes, if not drinking purpose. So they employ various treatment processes, as I mentioned already, to remove various contaminants from wastewater. Even aquaculture water is a wastewater, and from sewage. But there are inherent inefficiencies in all these processes. Their retention in the treated water gets disseminated into the environment. So primary treatment is usually not efficient. We have to use advanced treatments. It could be sand, and sometimes, some easy things are always there. And disinfection, as I said, and also chlorination, UV radiation, and ozonation. But there are now some cardinal studies on the use of biochar or on the use of, use literally, the problems associated with inefficient chlorination or ozonation, and how the genes thrive. Bacteria containing some of these genes, or the genes themselves do not get completely removed. So conventional activated sludge treatment that is done removes the fecal coliforms, enterococci, E. coli, and this is better in comparison to the septic tank systems. Now, in a study in 2015 done by Mao et al., also by Yuan, as well as another study by Souza and Zhang, different studies, Now, what they have shown is the disinfection steps like chlorination, UV radiation, and ozone alone, in combination with each of these methods, they were applied to assess the efficacy in removing the bacteria as well as the genes. Now, the results are startling. It's not able to altogether remove the antibiotic-resistant gene. Some of the cleaning methods, they increase, in fact, the proportion of the bacteria through the alteration of the cell permeability by activating. We know there are different mechanisms, with efflux pumps being one of them, which was actually designed by a bacteria to remove a lot of toxicants that come into them. And therefore, these toxicants that are used during the process of treatment also are removed, because the will to survive in any microorganism is so strong that they would use all kinds of mechanisms to get rid of any toxic material or chemical that is used. Then accumulation of intermediate disinfection byproducts. All of this promotes the horizontal gene transfer, particularly where, as I mentioned before, when there is prolonged contact with low chlorine concentration. This is true of even antibiotics. That is why we are always told, okay, to complete an antibiotic course, antibiotics are loaded, but we know many times in our country, off the counter, over the counter, you can get antibiotics and you take it maybe for a day, and these low levels that persist, though you may have recovered from the problem, tend to make you more susceptible to problems in the days to come. Now, one of the sustainable green wastewater treatment options that has been considered an alternative to the wastewater treatment plant as a component of conventional method, which has the ability to remove antibiotics, the bacteria, as well as the gene, are the wetlands that get constructed. These, what are wetlands? They are small semi-aquatic ecosystems, and a great population of different microbial communities thrive there, multiply, and various physical chemical reactions happen, and all of them are seen to manage the agents for antibiotic resistance that may be present. Another study by Alexander in 2016, of course, co-workers are there. They have shown that chlorination results in the survival of the bacteria, and there are different susceptibilities to disinfection. We carried out these studies in our laboratory. Also, I know many centers in India are doing even better work than whatever I'm quoting, because I'm aware a little bit of what we were doing. It may be unpublished material, but they strive to, be presented in a talk like this, because any talk is a food for thought. So the ozone treatment causes DNA damage, but has a reduced effect on the guanine-rich DNA. Ultraviolet light is absorbed by organic and inorganic matters that are present. So we know one of the primary ways in which wastewater treatment should happen is removal of all of the organic and inorganic matter, the sludge, which is to be removed before treatments take place, because the damage that is brought about by the UV would be the damage that is done to the bacteria would be directly proportional to the removal of all the organic and inorganic matter. Additionally, see, there are different sensitivity of the bacteria to UV irradiation. Laboratory studies have in different people shown, because we also know that, see, the cell wall penetration by some of these radiation matters. So we have sporulating organisms, we have organisms that are gram-positive with thick wall, we have gram-positive, say, mycobacteria that have got teichoic acid, and then we have so much of variation in the ability to penetrate, and therefore that the basic research becomes very important in laboratories in order to add value to whatever processes are being planned. Now, biochar, one study by Ye et al. Have shown this active charcoal that has been derived by pyrolysis of carbon-rich biomass has rich mineral elements. It's highly porous, and therefore this large surface area enables all the antibiotics that are present there, the resistant bacteria, as well as the genes that are present, to be removed. Now, if we look at different sectors, it is not meant to talk about it, but just to let us know, we may use poultry waste for fertilization of the pond. We know sewage-fed aquaculture ponds are there in West Bengal. We are aware how cow dung is used for fertilization. Now, when all of this is done, of course, we would like to call it organic production, because we are not adding any artificial material to fertilize, but at the same time, we must remember the problems that would come from them. Now, there are many, many pathogens, and there are even more antibiotics that get to be used. A cursory glance at all the antibiotics would show you how these very antibiotics we must have seen in the prescriptions when we have serious problems, especially as empirical treatments. So the diseases that are caused by them, the antibiotics that are used, they must really raise eyebrows, because for some which are very common pathogens like the methicillin-resistant Staphylococcus aureus, we find the use of vancomycin, and today we are talking about vancomycin-resistant Staphylococcus that is coming in the way of treating humans. We are also aware of the anti-tubercular treatment rifampin, and all of this, we have looked only at aquaculture, yes, but we did not look at other sectors that can contribute to the aquaculture ponds also carrying these antibiotic residues. And we need to be very, very, therefore, focusing on educating all the sectors. Look at the tuberculosis causing organism. And if we see, Mycobacterium is, yes, very serious. And if we look at the drug that is used, clarithromycin, rifamycin, it is shocking. And not to speak of all the other third generation cephalosporins, carbapenems, aminoglycosides, and quinolones that get to be used. Let us take the sector of aquaculture. We are not far behind, because everything that is done with terrestrial animal culture extrapolates to aquaculture. We have learned everything what they are doing, forgetting that the terrestrial environment and the aquaculture environment are different. In the terrestrial environment, when it is injected to an animal, or if it is given through feed, or if a human is given a particular medicine, we are aware that the drug reaches the site, reaches the cultured animal, then human being. But in aquaculture sector, we are never sure. When we add to the feed, it reaches to the environment, and we cannot catch each animal and inject them. So the problem is actually very acute in aquaculture when we dump methods. And are we aware of the excipients that are present associated with all of these drugs? So when we look at these names, as these slides are shown, apart from hearing me, you are looking at the names. Well, I'm quite happy if any of you want to later know a little more about some of these, you can always email us. But some of these kinds of studies that we are also doing shows that we are really at a very serious stage of not having any antibiotic available to treat us. So yes, warm water fish, cold water fish, drugs that are used, different people use, and therefore it would be really very, very naive on our part to say that antibiotics are not used. I'm not talking about India alone. This is the world situation. Some countries are more educated, and we are aware where Norway antibiotics are banned for salmon culture. With same Norwegians, they do culture in Chile, and a lot of antibiotics are used there, which are brought into this. So Norway has managed their environment through vaccination without antibiotics, but the same companies are selling all their antibiotic also in Chile. So we need to have a view that the whole world is a village, and we are all living with the same risk worldwide. So aquaculture, yes, cold water, warm water pathogens. Then livestock, again, all the antibiotic names, if you see, it will shock you. So being in a health university, for me, I'm facing these kind of antibiotics, and there is studies that are going on here from patients. But we are also the victims of what have been used in other sectors. So piggery as well, and zoonotic diseases, close contact with animals, vector-borne diseases, foodborne illnesses, airborne disease, waterborne disease. All of this have to be treated, and very often there is empirical treatment that is done without being able to weigh. There are many factors that increase zoonotic emergence. I will just skip through some of these. Now, if you look at the Israeli desalination and wastewater treatment, which becomes a global model for water scarcity, now you can see that in Eilat, this sewage utility says all of the country's wastewater that is treated and transferred to the last drop to farmers who use it to water crops. Now, we know agriculture, in spite of being actually locked with saline waters everywhere, yet this country has the best of technologies available. 80% of the wastewater flows back into the ecosystem without being treated or reused. This is in all other parts of the world. But look at what is happening in Israel. So, reverse osmosis, seawater desalination program has enabled Israel to be a leader in water recycling. So necessity is the mother of invention, and when they don't have fresh water, they have to resort to technologies to see that their health as well as the environment is taken care of. The United States Environmental Protection Agency was founded in 1970, and we find that it was created to protect the people, the environment from all the health risks. A lot of research is conducted, and environmental regulations that are stipulated are very, very strict. Now, their wastewater research includes development of methods for detecting and treating the contaminants, assessment of antimicrobial resistance in wastewater. And of course, during the pandemic outbreak, they were working on developing methods to monitor the municipal wastewater for the SARS-CoV-2 virus, as well as other contamination that found their way, contaminants. Now, the alternative wastewater disinfection that they use is very important. Now, the UV exposure or application of disinfectants and use of UV especially can be very energy intensive, and overuse they have found of disinfectants can be harmful, with chloramines forming, due to the reaction of the chlorine byproducts with the protein material that is there in the environment. So the alternative that they found is the peracetic acid. This is known to reduce, of course, health and ecological impact due to the continuous use of chlorination. But to increase the effectiveness, combination of peracetic acid with the UV technologies is being tested in a very, I should say, sustained manner. Now, what are the specific areas of focus for the EPA, the Environment Protection Agency, with regard to antibiotic-resistant bacteria and the resistant genes group of contaminants? So, assessment of relative contributions of hospital and residential wastewater flows to antimicrobial resistance that is there in the sewer networks, characterization of the resistant bacteria and genes in the biosolids, development of a PCR panel for the broad range analysis of the genes related to fecal pollution in the wastewater, assessment of the bacteria and gene loadings on wastewater treatment plants using molecular and cultivation-based assays, and improving bacterial concentration methods. So the dynamics, now coming to UK, I took some of the advanced countries because the lessons we can take is from there, and water being a factor common to aquaculture world over. And if we have to make water recyclable, available for aquaculture, we need to also adopt whatever is most practical for us. So Darwin, this is the dynamics of antimicrobial resistance in the urban water cycle in Europe. This was one of the first projects to perform a cross-European examination. So they looked at the fate of the resistant bacteria and the resistant genes that were resulting from discharged hospital and community waste, and finding its way to all of the water bodies. Years ago, I know about 40 years ago, I think 30 years ago, Dr. Otta was still in my laboratory where we were looking at methicillin-resistant genes, consequent to a paper that had appeared in Germany, where a hospital wastewater, which flowed through a river, which flowed through 3 countries, was carrying, and at every stage it was monitored. And they had at that time the molecular tools to see whether it was the same group of genes, and they tracked it. So traceability is also very important, because we know when we talk about, say, tetracycline resistance, there could be many mechanisms. You could look for the TET genes. You could also, a human may have tetracycline resistance that comes about, and it may be from an aquaculture organism that had also tetracycline resistance. And when being told one is the culprit and one is, of course, the victim, it would be good to see the mechanisms in the laboratory. And it has been found that the mechanisms to a great extent also decide the source from where it was obtained. So we can prevent ourselves from being unnecessarily blamed. It could have come from any other sector as well, because you could have the efflux pump in play in one sector, you could have the genes playing a role in another sector. And therefore, molecular tools do become very helpful. So to assess the risk of these resistant genes returning to humans due to environmental exposure, the goals were set for Darwin. And to guide the risk assessments, they made a predictive dynamic mathematical model for the urban water systems. And this they expect will help to assist health and sewage management system. Again, this is an urban water cycle on one side, and what are all the various treatments that are happening. Exactly from the previous slide, this is a spillover. Now, there is another technique, Jokasu technique from Japan, and IIT Rourkela validated this and adapted this technology. It is based on like for the hilly regions, they use this technique. So what was very important here is, as compared to septic tanks that we are very familiar with, where anaerobic treatment is most important, in Jokasu technique from Japan, you have anaerobic treatment and aerobic treatment going on parallelly. In the septic tanks, the retreatment of the treated water is very essential, even for releasing to the land-based use. But in Jokasu technique, this treated wastewater can be directly discharged to the environment. These images, of course, explain greater detail the differences between what happens in a septic tank and what happens in that Jokasu technique. So zero liquid discharge can help contain environmental AMR. Now, the zero liquid discharge, when we say, this is a kind of treatment process that is designed to recover and recycle all the liquid waste from an effluent system. So I just mentioned about the UK, where it's almost recycled 7 times and then discharged finally. So the treatment is so efficient to make the water highly potable for human use. Forget about all of the other uses for agriculture and so on. Now, pharmaceutical company in Gujarat has designed a zero liquid discharge unit to remove antibiotics from the manufacturing wastewater. So I'm coming towards the end. So when we talk about prevention and control of the issues that we have talked about, see, there are several drivers we have already seen: clinical, biological, social, political, economic, environmental. So all of these are drivers to antibiotic resistance development, and the dissemination happens to all the sectors. So our first lesson would be use less antibiotic. We cannot do away with it because we are aware, we are dependent. But what to use, when to use, how much to use becomes very, very important in deciding how we manage serious problem globally. To mitigate public risks from livestock waste or from human waste or from in-farm reduction, how we can proceed. See, just I think day before yesterday, there was a very major seminar from the FAO, from the in-farm group, and there they very nicely explained, because that was with specific reference to aquaculture, and other sectors were spoken by others. And it was, for me, very exciting to hear, because all this happens between 9 o'clock and 11 o'clock. We just go to the sites and see there is a talk and we start, attending them. So maybe most of what I've spoken is already contained in this particular slide. So with the World Antimicrobial Awareness Week being celebrated, we need to celebrate this the year long all over the world, with developing countries being more responsible and handling antimicrobials with care. Well, on a lighter note, you can see, of course, it says teacher and me, but it's a policeman and maybe a public. So what is the opposite of antibiotic? The uncle antibiotic. And now you can see in the second slide, the viral flu organism is just telling the public, see, you're using azithromycin for a viral flu. I told you, see, they make other bacteria resistant even though it was a viral flu. And you can see the picture of a bacteria is looking like a human. Yes, bacteria, when you treat with antibiotics, yes, they become very, very, very weak. But when you don't complete the course and don't really load it in the first instance to kill, they become resistant. And lastly, you can see it's maybe a kitchen area. You can see one bacteria telling the other, you want to be a superbug? Stick some of this into your genome, and there it is, the transmissible elements like a plasmid, it could be a transposon, it could be an integron. So this just spreading it. So they just talk about, and one bacteria telling the other, don't worry, no antibiotic can affect you once you take this up. So it was on a shortcut through the hospital kitchen that Albert, well, it's a West country, so they named the bacteria Albert. But in India, it could be anything, Ram, Lakshman, so on. When it was first approached by a member of the antibiotic resistance. So I thank you for listening, and thank you for your patience rather in listening. It's not really, I don't know how much it was relevant to the antibiotic awareness week, but I think there are too many things that need to be known for all of us to take any concerted action. Thank you very much to the organizers for this opportunity.
Thank you, madam. Thank you very much. So, do you have any questions?
You're welcome.
From the audience or from the online? If not, then thank you, madam, very much. We are really fortunate to have madam with us today. So if I go on telling about her credentials, it may take another 1 hour. So in short, to mention that she did her MSc and PhD in medical microbiology. Then initial career she developed in some of the famous medical institutes like Jeepnor Pondicherry and KMC Mangalore. And then she joined Mangalore Fisheries College, and from where she retired as a professor and head of the microbiology division. So before retirement itself, she was the brainchild in developing a biomedical science institute at NITE University in Mangalore, where she became the founder director. And at present, she is continuing in the same institute as director of projects in the DST scheme. And additionally, she is also the director of UNESCO mission and also director of FAO AMR referral laboratory. And she has been postdoctoral fellow and UNESCO fellow and visiting scientist to numerous famous institutes in different parts of the world, like USA, UK, and Netherlands, and that list continues. I cannot tell so many, I don't remember also. She has also numerous awards to her credits, like Best Scientist Award, Best Women Scientist Award, Young Scientist Award from numerous organizations. And she has also Rafi Ahmad Kidwai Award from our ICR organization. And she is also fellow of different organizations like NAS and so many other things and all. So with so much credentials, really we are lucky to have got madam today. She is extremely busy, but somehow I requested her to give a talk for our staff and others. She was agreed. So we thank you profusely, madam, for accepting our invitation and delivering a talk today, such a beautiful talk. It was very educative, and I am sure many of the people, they might have got benefited by your talk. So thank you very much, madam.
Thank you, and all your colleagues, because I know it's a very long talk, but when you are
it was exactly 1 hour, madam. It was very nice.
Thank you.
Thank you everybody for joining with us. Thank you all.
I am known for not keeping time. That is why I have to start apologetically and also end.
No, madam, perfectly. It was 1 hour, less than 1 hour.
Okay. I'm improving. Thank you.
Thank you, madam. Thank you.